Hello, good evening everyone. Welcome to today's clinical grand round. We are speaking on molecular epidemiology of bacterial respiratory pathogens, pre-COVID and post-COVID era. As all of you know, the last 3 years have been really very terrific for all of us. We have actually faced pandemic of COVID-19. Then there was fear of monkeypox. Also in between we heard about influenza, even influenza cases. So most of the time the focus of all the microbiologists and of all scientific fraternity has been towards viral infections. And some rare bacterial infections got neglected. Although cases were of viral infections, we were still treating the patients with antibiotics, and we did see a lot of emergence of multi-drug resistant strains. And that is where, the epidemiology of bacterial respiratory pathogens comes into importance. So we have realized that during this period, many of the laboratories were either not looking for respiratory bacterial pathogens or were taking any case coming to it as viral infection, or otherwise it was happening that even if the infection was there, somehow it was overshadowed by other viral infections or viral respiratory pathogens. So we thought in today's clinical grand round we will focus.
Thank you, Dr. Rama, for the truncated version. Uh, as you said, the bacterial respiratory pathogen, as you realize over the next 1 hour, has gained a lot of notoriety during the last 2 years, specifically due to COVID-19, as well as influenza, swine flu, and porcine flu, etc. Today's topic we're going to discuss in two halves. First, I will talk about the pre-COVID era, basically focusing on pneumococcal infection and molecular epidemiology, and a little bit about Haemophilus influenzae, the new variant of Haemophilus influenzae infection, that is the non-typical Haemophilus influenzae. Can I have the next slide? Okay. Uh, just to give a very brief introduction, this is a disease of the low socioeconomic condition, mostly developing countries in Asia and the African nation. In fact, you'll be surprised. Bangladesh has much more, as far as the health is concerned, they're much more advanced than us. Therefore, as I said, that low economic and middle income countries like the Eastern African countries, the Western African countries like Algeria and the Kenya, uh, uh, Congo, etc., Ghana, these areas, Nigeria, Niger, these are the areas where you have majority of the respiratory tract infection. And respiratory tract infection, you realize it targets mostly the children below 5 years of age. Below 5 years, and 50% of the mortality below 5 years, 15% of the mortality below 5 years is because of the bacterial respiratory etiology, mostly strep pneumoniae as well as Haemophilus influenzae, coronary bacterium, diphtheria, Bordetella pertussis, atypical pneumonia causing etiological agent. As you realize that the severe form of the pathogens, we have an excellent vaccine. So ideally, these organisms should not be the cause of mortality, even in a developing country. But as you understand that because of the low socioeconomic condition, vaccine for these primary pathogens causing lethal infection are not available to all and everyone. That is one of the reasons. And I will not go into the viral etiological agent, as you already realized that COVID-19, before the SARS as well as MERS, and influenza. In fact, I was hospitalized with influenza, atypical pneumonia, H1 and N1, and it was diagnosed by the molecular diagnostic assay because that is the mainstay of molecular diagnosis. Just briefly, these are the etiological agents, but today we are going to talk about the acute infection due to Streptococcus pneumoniae, Haemophilus influenzae, and subsequently chronic infection due to uh, Mycobacterium tuberculosis. Just a brief understanding, very briefly, we need to understand the background immunology of respiratory infection. You realize that the immune system, both the innate immune system as well as humoral immune system, is in the peripheral vascular system is very, very robust. So a very robust innate immune response as well as adaptive immune response, including humoral as well as cell-mediated immune response. Like the CNS, central nervous system, where the immune system is supposed to be rudimentary, it works at least say 1/10 of the level that it works in the CN and the peripheral system. Similarly, if you compare the upper respiratory tract and the lower respiratory tract, you will realize that upper respiratory tract with adenoid glands, Walden's ring, tonsils, and large number of lymph nodes around the oropharynx, we have a robust mucosal immune response along with the cell-mediated immune response. Just like the mucosal immune response that you have in the intestine. The patch patches with diffuse different kind of lymphoid tissue as well as localized mal or mucosa associated lymphoid tissue. So a very strong immune response is induced in the gut as well as in the upper respiratory tract mucosa. We do not understand very clearly how does the respiratory tract work in mucosal immunity induces, but we understand that if you give an intranasal vaccine for influenza or any other respiratory tract infection, it induces a very strong mucosal immune response. Therefore, COVID-19 against COVID-19, we have now a licensed vaccine for intranasal immunization. The story at the lower part of the the lung is or respiratory tract is very different. If you look at the anatomy of this is a paper that we published in the International Review of Immunology, an update on host immunity correlation prospect of reinfection COVID-19 in 2022. If you look at the anatomical structure of the alveoli is lined by a very single cell epithelium, which is specialized during the evolution to exchange gases, get oxygen from the blood and release carbon dioxide from the tissue. And this is done by the very thin capillary that is associated with the alveoli. Difficulty of this is that unlike the upper respiratory tract infection, upper respiratory tract mucosa, the lower respiratory tract in the alveoli, the amount of immune cells that you find are very, very small, a few number of antibodies and a few macrophages, pneumocytes. These are all what is you have in the alveolar sac. So if you compare to the upper respiratory tract mucosa and immune system, in the lower respiratory tract mucosa, the immune system is very, 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 very low. So it is the immune privilege site. If the organism like COVID-19 virus or influenza is able to enter through the trachea, land up in the lower part of the respiratory tract, and if it is new virus, mutated or natural virus or bacteria, it may actually cause devastation because it induces a, for example, pneumococcus and pneumolysine, autolysine, it causes a severe inflammatory response in the alveoli and causing the inflammatory cells to pour in, causing red hepatization and subsequently the individual partial oxygen pressure inside the alveoli or the blood goes down. So from 95 above, the PSO2 will come down to probably below 80. That is one of the reasons why these individuals have a severe morbidity as well as mortality. Okay, this is as far as the understanding the host immune response. As I told you, we do not understand how the mucosal immune response work in the respiratory tract, but we do understand that you need both the mucosal immune response, IgA, as well as IgG, systemic immune response, to protect you against COVID-19. Therefore, a DNA vaccine or RNA vaccine that is given in in COVID-19 will induce, because we understand that DNA or RNA molecule, if you inject into the cell, will induce both the cell-mediated immune response as well as humoral immune response. Probably in the upper respiratory tract infection, so lower respiratory tract infection, both the humoral immune response as well as mucosal immune response as well as cell-mediated immune response is very, very important. Just to define what is the molecular epidemiology. The term molecular epidemiology was first coined by Edwin D. Kilbourne in 19. Edwin D. Kilbourne was the person who actually invented the influenza vaccine. He cultured large amount of different strain of influenza and he's given the vaccine to the world. So he was the person to understand or describe the molecular epidemiology in relation to the influenza virus. Subsequently, Janice S. Dorman, she's an epidemiologist working with the Pittsburgh University in the US. She defines molecular epidemiology as a science that deals with the contribution of genetic and environmental risk factor identified at the molecular and biochemical level to the etiology, distribution, and control disease in families and population, both in the community as well as in the hospital. Simply put, we'd like to say that is a discipline that uses molecular diagnostics tool to trace the development of disease and understand the environmental factor, epigenetic factors, as well as virulence properties of the organism that contributes to the disease. Okay? Very quickly, classically, microbes or pathogens that cause the human disease or animal disease are classified by the standard assay biotyping based on the phenotypic characteristics, what kind of sugar, what kind of cell wall structure they have, etc. Or fast typing using bacteriophages. So it can be biotyping, serotyping. But all this system, the difficulty with this system is that they may not actually identify different organisms because different organisms may carry the similar properties. Some may be, for example, even if you do a serological typing, two strains may have a similar kind of epitope, so they may share. So ideally, these may not answer whether these strains are exactly the same or they're different. So we need something much more defined or much more uh, clarity in understanding a strain. So if you understand in 1953, Watson and Crick first discovered the DNA, and that changed the whole of the diagnostic field as far as the infectious disease is concerned, as far as the genetic diseases are concerned. In the in the early 1980s, 83 to be specific, Carey Mullis first invented the polymer chain reaction. So during the last, in the 70s, 80s, and 90s, the whole of molecular pathogenesis or the molecular characteristics of the virus have been completely analyzed. So now we have a large number of different molecular tools that are routinely used as a diagnostic purpose, for epidemiological purpose, as a for vaccine preparation, etc. In fact, the impact of these molecular tools in the preparation of the vaccine in the last COVID-19 vaccine preparation, within 1 year, you realize that whereas it took us 20 years to make a vaccine, in the COVID era using molecular tools, this waiting period from 20 years, it has been reduced to only about 1 year. So within 1 year, we have a vaccine for COVID-19. So what are the molecular assay? Restriction fragment length polymorphism. That basically tells you that almost all the pathogenic DNA human genome has certain unique sequences that can be utilized to identify the organism. So all of us carry, including the bacteria, including the virus, carries unique sequences, linear sequences, randomly repeated sequences, tandem repeats, etc., or insertion elements which are very, very specific to a part. So utilizing those information, one can identify the bacteria or pet virus, etc. And in addition to this RFLP or restriction fragment length polymorphism, you have hybridization assay, northern blot for the RNA, southern blot for the DNA, randomly amplified PCR product, amplified polymorphic DNA, pulse electrophoresis if you want to look at the whole genome. Then subsequent, these techniques are very laborious. It requires very calculated, expensive equipment, plus from individual to individual, from laboratory to laboratory, their reproducibility may vary. Therefore, you need something much more specific. So you have sequencing. You sequence the whole genome compared to, because if you compare the early 90s where you have the applied biosystem looking at a genome, looking through the either through the gel sequencing or through the capillary sequencing, it is to take days to complete the sequence of a virus. Now what we do is you can do the whole human genome sequencing probably in a month, whether it took us about 5 years to complete the whole genome sequencing. Then subsequently we realize that sequencing the whole genome of a bacteria virus may be not worth it because it is very expensive. So what you do is you take selected sequences of the gene of that individual pathogen and subsequently sequence those short fragments of DNA, whatever DNA we are talking about. So you sequence those and then compare these sequences from on the isolates from different parts of the either world or of your locality. If you want to be more specific, that if you want to identify strep pneumonia isolates or a virus, let's say herpes simplex virus from Delhi or from Assam, then you will look at the specific sequences because ideally the whole genome will be exactly the similar. So you want to see if there is a specific mutation. For example, the the Omicron variety of the COVID-19 and the gamma beta variety or alpha variety of the, when they wanted to see why the Omicron variety was much more rapid as well as much more less virulent, then we looked at the mutation. So single nucleotide polymorphism is a technique which looks at the particular change at a particular position, single mutation. So these are the techniques that are currently used in molecular epidemiology to identify what are the application as far as the infectious disease is concerned. I'm not talking about the general genetics or otherwise. Application of molecular epidemiology as far as the infectious disease is concerned, identify, characterize pathogenic isolates or identify newer species for COVID-19 or influenza virus that attacks every year, the pandemic variety. Comparison with the existing member, you can look at the strain from here, strain from Japan, strain from the other areas, identify new variants. Example for a COVID-19 variety, alpha, beta, Omicron, variant of interest variant. So you do a sequencing of the whole genome because now you have the whole gamut of technology that looks at the sequence in a very short period of time. So in fact, these days you have a tabletop centrifuge that quickly looks at the genome sequence in a very short period. So hundreds of strain you can sequence and look at the variation. That's one of the important factor for disease surveillance. Tracing pandemic, again, good example is influenza virus, H1N1, COVID-19, Omicron variety, outbreaks of epidemic. If there is an outbreak of Acinetobacter, AIIMS, you want to compare the one of the others. So you can identify the genome and then compare. Identify prototype candidate vaccine. If you sequence the whole genome, you will know whether this particular strain is suitable for you when you compare a line with the virus with a different virus. For example, I'll just give you a good example. When you looked in the early 90s, we were working on the rotavirus vaccine. We're looking at the genome of the rotavirus. Rotavirus is a segmented virus, 11 different segments are there. And we were looking at the two specific genes, gene 4 and gene 9, which codes for BP7 and BP7 and BP4, two important neutralizing proteins of the virus. When we look at the genome of the virus, we found that India, unlike the West, in the West, the rest of the world, this is a very highly conserved virus. You have only serotype 1, serotype 2, serotype 3, serotype 4, along with the P842, two different varieties of gene 4, and you have only 4 types of serotype G serotypes. But when you look at the India, you will find that India has a huge variety of diversity. And this diversity helped us to identify a strain in our nursery, AIIMS neonatal strain 116E. And this neonatal strain 116E that we characterized in the early 90s, and based on the molecular epidemiology of the virus, we have been able to identify this virus, completely characterized by virus by doing the sequencing. And this particular 116E finally, I think 2016, this has been licensed as a vaccine for Indian scenario. So this is how molecular epidemiology helps you to characterize, make a prototype vaccine, and then subsequently you see, and this whole procedure has taken 20 years. But you know that with using molecular methods, we're using fast track legal structure procedure. You can make vaccines as given example for COVID-19. Within 1 year, you can make a vaccine for COVID-19, whereas this particular vaccine has 20 years, phase 1, phase 0, phase 1, phase 2, phase 3, and then vaccine was licensed. Now, the phase 4 is going on. I've been in recent Lancet, the phase 4 of the rotavirus vaccine, 100 Rotavac, has been published. As a microbiologist, as an infectious disease person, my laboratory at the Department of Microbiology is involved primarily HIV immunology and immunology, but we're also interested in the host-pathogen understanding of the different respiratory tract infections. So we mainly focus on strep pneumonia as well as Haemophilus. It will not be possible, but very briefly I will like to say that we are involved in serosurveillance of strep pneumonia in Delhi and NCR region, molecular characterization of PSPF, pneumococcal surface protein. I'm going to describe a little bit more in detail. And then subsequently, invasive, we want to look at the difference between an invasive as well as non-invasive pneumococcus that colonizes the throat and those strains that are isolated from the sterile body fluid. Okay, so multilocus, we now understand that multilocus sequencing is one of the probably most promising technique which is reproducible, and there is a data bank available where you can align your strain along with the genes and you can give a newer sequencing type. Lastly, we are also working on the non-typeable Haemophilus influenzae. I have 1 or 2 slides to show. Just to say that Haemophilus influenzae has been completely, type B Haemophilus influenzae, which is the one that causes acute bacterial meningitis, has been more or less eradicated from the developed country. And also, it has been this HIV vaccine, the conjugated Haemophilus type B vaccine, has been introduced in the EPI, Extended Program of Immunization. Therefore, type B Haemophilus influenzae as a major cause of morbidity, mortality, meningitis in Indian scenario or even the worldwide is almost diminishing. So in that place, non-typeable Haemophilus influenzae. Why non-typeable? Because it doesn't have a capsular polysaccharide. So the vaccine that works against the Haemophilus influenzae type B doesn't work against the non-typeable Haemophilus because non-typeable Haemophilus doesn't have a capsule. Very briefly, to tell you, strep pneumonia is still the major. This is the pneumococcus. So strep pneumonia worldwide, if you look at the disease profile, Delhi or disability adjusted life year per 100,000 population, you realize that strep pneumonia is still a major cause of public health issue. So it causes maximum mortality, morbidity. Therefore, this is something we need to tackle. We need to have a good vaccine. And you will understand that this is This is a pathogen that works mostly in the children, that the children below 1 year are the ones who are actually target of this infection. And unfortunately, all the polysaccharide vaccine, because the children below 1 year, their immune system is not matured, polysaccharide vaccine doesn't work below 1 year in both against both Haemophilus influenzae as well as strep pneumoniae, because both these carry capsular polysaccharide. So any capsular polysaccharide vaccine below 1 year will not work. So you need a conjugate vaccine. In a serosurveillance study that we carried out, what we found that this has been published in PLOS One in February 2020. Thanks to Sambuddha for persisting working on this. So you realize that the children, infant below 1 year has the maximum number of asymptomatic carrier, as well as they have a lower respiratory infection. So out of 536, you almost have 25% of these individual has symptomatic infection, probably due to pneumococcus. Asymptomatic infection, about 10 to 15% individual will carry staph pneumoniae. So even in India, I told you the picture for the rest of the world. Staph pneumoniae is a major cause of mortality morbidity, even in Indian scenario. You know, uh, capsular polysaccharide based classification for pneumococcus divides the strains, strep pneumoniae strain, into more than 90 different serotypes. So if you have a vaccine for pneumococcus, the vaccine can only contain or consist of a certain number, limited number, say 23 capsular polysaccharide vaccine, or 13 conjugate vaccine, 10 conjugate vaccine, 7 conjugate polysaccharide vaccine. So these limited number of vaccine containing serotypes will not work for the 90 different serotypes. So what is the major issue? Major issue is that because there is a large, more than 90 different serotypes. Okay. So at one point of time, this particular study was carried, as I told you, some other study, and we found that there are more than 40 different capsular polysaccharide serotypes circulating in India, and majority of them are causing asymptomatic, and some of them causing the blue color asymptomatic infection. So as far as the difference is there concerned, the phenotypically there is no difference between a asymptomatic bacteria as well as invasive bacteria. So the same organism can colonize your throat at some point of time when host immunity is done, specifically in elderly, where there is smokers, alcoholics, diabetic, in those individuals, they will take the opportunity and become invasive. So this is one of the study that look at the serosurveillance. In a subsequent study carried by Shefali at her hospital, this is a hospital-based study. So you can see there are about 19 different serotypes circulating at one point of time. And these colors defines the invasive, non-invasive, locally invasive, as well as asymptomatic strain. The same serotype can be invasive, it can be locally invasive, or it can be as a commensal. So practically, we do not see any difference between their genome, and that will be confirmed. So first molecular study, epidemiological study that we carried out for pneumococcus is trying to characterize this virus, the bacteria, in terms of finding a candidate vaccine. And what is the candidate vaccine? Uh, well, before that, let's look at what are the currently vaccine that are available for pneumococcus. Basically classified into two groups. Simple polysaccharide vaccine, you have multiple different serotypes, so you take fragments from each of the bacteria, 25 microgram from serotype 1, serotype 2, serotype 3, and pool them. It's a cocktail of polysaccharide capsule in terms of weight 25 microgram, and pool them, make a vaccine. So 23V valent vaccine has 23 different polysaccharide capsules. Similarly, then we understand this vaccine doesn't work in children below 2 years of age because their immune system is poor. So you conjugate it with either tetanus toxoid or diphtheria toxoid or outer membrane protein and make them, if you take 7 serotypes, 10 serotypes, 13 serotypes, and conjugate it with a protein molecule. This will work very well in children below 2 years. So these are the currently available vaccine, but you have only 23 maximum number of polysaccharide the serotypes in a particular vaccine, or 7, 10, or 13 valent vaccine. So limited number of serotypes are covered. So at some point of time, if you give vaccine insistently to a population, in those situations, the vaccine serotype will change. A new serotype of vaccine will replace the community and they will cause disease. Therefore, what you require is you need to look at the capsular polysaccharide, because conjugation process is very, very difficult. It's a very complicated process. You can conjugate so many numbers, 13 different conjugate can be conjugated. But you cannot conjugate 40 strains, or you cannot conjugate 90 strains to give the whole of the expanse of the coverage. So what you need to do is you need to look at the alternative vaccine. So what are the different alternative vaccine? I'll just take a, so you have a candidate vaccine called pneumococcal surface protein A, or PSPA. Uh, what is the advantage with this? This is a common protein, most potent vaccine candidate. It's a surface protein present in all pneumococci. It is cross-predictive and it interferes with the complement. So then we looked at the this particular protein has an alpha helical region, and based on the alpha helical region, this can be divided into 3 families or class subclades, 6 different subclades. Why do you need to group them? Because we need to find which strain of this pneumococcus. So all pneumococcus can be classified into these 3 groups. So this is a common protein. So we can pull the family 1, family 2, family 3 together and we make a vaccine. So what we did was, using the PCR technique and these primers, we looked at invasive strain as well as nasopharyngeal strain, asymptomatic strain, and defined the clades. So we found that in our setup, we have family 1, family 2, no family 3. What are different clades? We found that except clade 6, we have all the different clades. So this was a good candidate vaccine. This is as far as the commensal bacteria is concerned. Similarly, this is the circular dendrogram showing that in our setup we have, this is a reference strain, clade 6. So we have family 1, family 2, clade 1, 2, 3, 4. This would have been a good candidate vaccine. So similarly, when you looked at the invasive strain, we did not find any clade 6 or family 3, but we found all this strain. But the major issue with this strain was that PSPA, later we realized that it induces autoantibodies. So you don't want to give a protein which may induce autoantibodies, subsequently lead to some kind of a reaction leading to autoimmune disease. So that's why the PSPA protein was dropped. Her next step was looking at the multi-locus sequence typing and trying to identify our strain and look and compare to the rest of the world. Urvashi, I have just 2 or 3 more slides. Again, we realized that the best way to looking at the molecular structure of or molecular epidemiology of any strain is multi-locus sequencing typing. Very briefly, what you do is, in any pathogen, there are some housekeeping genes, which housekeeping genes basically they encode or code for certain enzymes which are necessary for the life of the bacteria. Without them, they will not survive. So you take 7 or 9 of this housekeeping gene, amplify with the specific primer, PCR products are produced, and then you do a sequencing on this product. Subsequently, based on the sequencing, these sequences are compared with the standard gene bank, MLST gene bank, and then you identify allele assignment is there. If there are single mutation, so they will use a specific number. Again, gene 2, you have particular sequence mutations, they will have. So all these 7 members are put together and a new sequence type is given. So based on this, ST51 will identify a particular strain that will have this kind of mutation. So MLST is a good way to identify strains of pneumococcus. So we found that in our setup, both invasive and non-invasive strain, we have 1, 2, 3, 4, 5, 6 different new sequence type that has not been discovered before. So our strains have certain number of Indian strain, a large number of diversity. Okay, just to show that these are the commensal strain and invasive strain almost lie with each other. So practically, if you look at the genome of this invasive, non-invasive, they exactly look similar. Lastly, my non-typable Haemophilus. I told you non-typable Haemophilus, non-capsulated Haemophilus is actually taken over from the type B Haemophilus influenza disease. So where one of my students, Sonu, I don't know if he's here. Yeah, so he has been working on this. We found that when we look at the MLST, same sequence, the 7 genes of the Haemophilus influenza is with them. First, we serotyped them. We found that these are non-typable. They cannot be serotyped with the existing 7 different capsular serotype A to F. We found that they are non-typable. And once we identify them as non-typable, we sequence the 7 housekeeping gene and then do draw a sequence. So we found that both the maximum number of this has been put in the gene bank and in the process of writing this paper. So now writing this paper. So we found that genetic variation was found to be highest in invasive isolates, while less common amongst the commensal and non-invasive isolates. So that was one of our. This is the same finding. Well, this is the reverse find. We found it in way back in 2011 when you look at the type B strain. Type B strain had much more conserved because the invasive strain will try to retain their genetic makeup. So type B Haemophilus influenza was more conserved, but here non-typable are much more diverse. Non-typable Haemophilus influenza it is now found to be common amongst bacterial meningitis as well as localized mucosal, say maxillary sinusitis or throat infection, especially in elderly where there may have already existing lung infection. Sorry, one more slide. Uh, I think that's the last one. Thank you so much, and I welcome Dr. Urvashi. I'm sorry I took a little bit more time than.
Good evening, all. So, with the beautiful preface having been given by Dr. Bimal, I'm straight away going to jump to something very interesting in the field of tuberculosis. I am only talking about this work which is currently ongoing under a BRICS study funded by DST. And so this is a collaboration between the BRICS countries. And that's why the several funding agencies insignias are here. I am the lead PI from India, while we have collaborators except from China. We have from Brazil, Russia, and South Africa. So I've kept the presentation very crisp so that the messages get through. Mycobacterium tuberculosis is known to be a clonal pathogen. By clonal, what I mean is the frequency of SNPs, what Sir alluded to earlier, is much less in the case of Mycobacterium tuberculosis. Also, horizontal gene transfer has not been reported in a large number of studies. There are only a few recent studies which does talk of horizontal gene transfer. Also, the M. tuberculosis, there are a lot of papers we published in the year as early as 2006 that there is a coevolution of M. tuberculosis with its human host. There were papers from Stanford around the same time which have actually delved upon this, and this is one concept that has been worked on. There are host-related selective pressures which have been postulated to drive the genetic diversity. And the genotypes of M. tuberculosis vary in their association with multi-drug and extensively drug-resistant TB. Hence, the relevance of molecular typing and molecular epidemiology for tuberculosis. I'll try and allude more later. So this is a figure which actually summarizes. So the molecular epidemiology tools that have been used for tuberculosis have been several. What we have used early on, so we started using molecular typing as early as 99 and year 2000 in our department. What we were using was called spoligotyping. You people must have heard us referring to it. But the studies in early 2000s used large sequence polymorphism as well as SNP polymorphisms to see if spoligotyping did match with the LSP and SNP genotyping methods. The nomenclature used here is basically the based on the LSP and SNP. So we have lineage 1, lineage 2, lineage 3, 4 and 5. So lineage 1, 2, 3, 4, primarily I have a nice map later. Yeah. So the lineage 5 and 6 are restricted to the west of Africa, and there is a recently defined lineage 7 which is restricted to Ethiopia. So this map will be able to, I'll be able to explain it better. Lineage 2, which is also called the East Asian lineage, is defined or reported more from the eastern parts of Asia, hence the name East Asian. China, Southeast Asia and parts of Russia have report this East Asian lineage 2. The spoligotype name for the same is the Beijing genotype. So we call it the Beijing family. Here we use the word lineage. So lineage 2 is here. Lineage 1 is called the Indo-Oceanic as per the LSP genotyping. It is around the Indian Ocean, south of India, eastern, southeast, eastern part of Africa, then parts of Southeast Asia. This is the Indo-Oceanic lineage or lineage 1. We have the lineage 3, which is primarily in India, and it includes, the northern part of India, and we call it CAS genotype or CAS family by this polygotype nomenclature. Then we have the lineage 5 and 6 in the Horn of Africa, and the lineage 7, like I said, is not depicted in this picture. It's been recently defined. It's restricted to Ethiopia, and then we have the, in the phylogenetic tree, the animal. This is where we can actually match the lineage 1, which was called the East Asian. Here it's called the East African Indian. This is the polygotype nomenclature. Lineage 5 and 6, like I said, is in the Horn of Africa. Lineage 3 is primarily in the north of India, the CAS genotype and polygotype. Lineage 2, the Beijing polygotype in China, Russia. Lineage 4 is, can I go back? Yeah, lineage 4 covers the most of the Western world. It has not been reported much from our country. And actually, if I could go on to the hypothesis of how TB spread, it will take another hour, but let me restrict to what I'm here to talk today. This slide summarizes the genotype-phenotype association that has been reported worldwide. So there is an association shown between lineage 2 or the Beijing polygotype and increased pathogenicity, increased virulence, and transmission of resistant forms of TB. There are other phenotypic differences between lineages, which include differences in vitro growth rates, hydrophobicity of different genotypes, the clinical outcomes, relapses, failures, treatment, retreatments, and then, like I said, Beijing is known to be more of an MDR. So those associations have been reported in several studies. Higher transmissibility of certain genotypes have been reported, and I'll again allude to this towards the end of the presentation. The host inflammatory response has been shown to be different for different genotypes. So again, this is the relevance of molecular epidemiology. High transmissibility of lineages 2 and 4 in comparison to lineages 3 and 1. So in India, we primarily have lineage 3 in the north and lineage 1 in the south, while China has lineage 2 and lineage 4 in the Western world have shown to be much higher transmissible. Now, amongst the Beijing, further sublineage transmissibility has been shown. The sublineage 2.2.1 has been shown to be more transmissible, though the others are more restricted. The our study was actually designed to study. So I think I'm not actually covering all of it, so I'll just restrict to now. This is what we published in the year 2007. Before this, the molecular epidemiology of tuberculosis in India was not known in such detail. There were few scattered reports from NIRT Chennai, which was a much older institute. And what we showed was from 8 centers in our country that the north of India, the samples were collected from Delhi and Lucknow. So in Delhi, we had predominantly, so this study, we only used polygotyping, while now we have moved on to the modern techniques, the other newer techniques. This here we showed the CAS spholigotype constituted 56.5% in Delhi, while the EAI was 22.7%, and the Beijing was 5.8%. In Lucknow, something similar, 44.6% CAS, EAI 41.5%, Beijing was 3.1%. Now, if we look at South, we had samples from Chennai and from Trivandrum. So Chennai had CAS only 11.5%, while East African Indian 67.5%. Beijing was 11.5%. Trivandrum had further lessening of the CAS. So if you see, there's a difference in the CAS. In the north is very high, EAI is less. Beijing is in the range of 6 to 9 throughout the country. This is in the year 2007, and now the epidemiology is changing. When we look at the south, and so in the western part, CAS is almost at par with what is there in north. But in the south, the EAI is much more, 89% in Trivandrum, 67% in Chennai, while CAS is much lesser. So we need to use very highly discriminatory markers, and that is why the LSV and SNP method I showed you. This is another picture of global MTBC phylogeny. And what I'm now coming to is what we have used for our study that I'm discussing today. We've used the SNPs, which are specific for the different lineages. So this is, this compares the LSV definition or the LSV lineage definition and the corresponding spholigotype definition and the SNPs that have been used for this study. Coming on to another slightly different aspect of TB infections. Now, what are polyclonal infections? Polyclonal infections are when multiple strains of M. tuberculosis are retrieved from an individual patient. Now, we started working on this about 6-7 years ago, and we published it in 2020. Polyclonal infections shown in varying percentage of isolates in different studies. Heteroresistance is nothing but a subset of polyclonal infections, that there is a difference in the resistance profile of the different clones present in an individual. So heteroresistance has an impact on TB resistance testing and TB treatment outcomes. So this is the PLOS1 2020 paper. We demonstrated that 32% baseline polyclonal infections in drug-naive TB. So for all these years, it was thought that only drug-resistant TB had polyclonal infections. But what we were able to show was drug-naive TB, that is sensitive TB, which has not been exposed to antitubercular drugs in the past, has almost 32% polyclonality at the baseline. 54% of these had resistance to at least one drug. There was a change in genotype detected while on treatment. Now, I'm only talking about rifampicin sensitive TB here. So this is what we noticed, and that's when we decided to look for polyclonality in further studies. Another paper, Nathavitarana, reported polyclonal infections to be positively associated with multi-drug resistance and other drug resistance patterns. So this is where I bring in the pre-COVID and the effect of COVID. I don't know if it's early to call it post-COVID, but at least the COVID did change the epidemiology of the M. tuberculosis in our country. So this is a picture of how it changed. So we are looking at the pre-COVID Mycobacterium tuberculosis strains isolated from North India and northeastern part of India. We have another study going on in the northeastern region where we're getting samples from Sikkim, Assam, and Tripura. So if you please see here, like I said, the Cas oligotype or the L3 lineage is the predominant genotype in most of the northern parts of India, followed by polyclonal infections, which is L3, L4 together, and the lineage 4. During the pre-COVID time, the monoclonal infections were about 67%, while the polyclonal infections were about 33%. Now, here, when we look at the post-COVID, this we are looking at the mixture of sensitive and resistant. During the post-COVID, or like I said, I may be, too rushed to call it post-COVID already. But yes, this is recent isolates during the COVID pandemic. What we found was a clear change. The L3, the numbers, due to lineage 3 were equivalent to the numbers due to polyclonal infections due to lineage 1, 3, and 4, and polyclonal infections due to lineage 3 and 4. So there was a distinct increase in the polyclonal infections. It went up to 71%. When we compare pre-COVID sensitive and the pre-COVID resistant, in the next slide I talk about post-COVID. The pre-COVID sensitive samples primarily were due to lineage 3, while the pre-COVID resistant samples were due to lineage 3 and lineage 4. Also polyclonal infections due to lineage 3 and 4. And this is to show the monoclonal and polyclonal nature. Amongst the sensitive, we had very few polyclonal infections, that is 13.2%, while 85% were monoclonal. Amongst the resistant, we had 47% were polyclonal, and monoclonal were 53%. Now, this after the or during the COVID pandemic changed. So amongst the sensitive, we had a representation of polyclonal this was polyclonal due to L1, L3, L4. L3 still retained a big chunk, but then we had polyclonal infections bothering us. Now, what is the actual worry about having polyclonal infections? Like I said earlier, polyclonal infections are more likely to go on to drug-resistant fever. And like I mentioned earlier, certain genotypes are more resistant to rifampicin and difficult to treat and may relapse. So these patients, though we may feel they're treated, they're cured, they've gone home, they may be lost to the system, and they may have a relapse, and they may come back with drug-resistant TB. Now, this tells us, so this is to depict the, purpose specified. Can you go back like this? Oh, yeah. So, a lot of graphs. Now, basically, now this was pre-COVID in Delhi. We had, lineages. So this is lineage 4. This is lineage 3. So individually, this was monoclonal infections due to lineage 3, lineage 4, and polyclonal infection due to L3, L4 during the pre-COVID time. And I will show you the post-COVID from the northeastern region. We do not have pre-COVID data for the northeastern region and the post-COVID data for the Delhi region. So this is what we found during the COVID pandemic, that the northeastern region had monoclonal infections 64%, polyclonal 36%. So what we observed was there was a distinct increase in polyclonal infections from before, which was 33%, and during the COVID pandemic, it went up to 71%. Salient increase in polyclonal infections and rifampicin sensitive TB from 14% to 72% during the COVID pandemic. Lineage 3, that is the Cas, which I showed you with the previous studies also. So like I said, the previous study was published in 2007 and the work was done from 2002 to 2006. So during that time till today in 22, lineage 3 has been the one which has been the predominant lineage. But during the COVID-19, we had equal number of polyclonal infections reported due to lineage 1, 3, 4, or lineage 3 and 4 in rifampicin sensitive TB. Amongst the MDR TB samples from Delhi, the lineage 3 and lineage 4 were responsible for equal numbers, while polyclonal infections due to lineage 3 and lineage 4 were responsible for double the numbers. In the northeastern region, the MDR TB samples were due to lineage 4 or mixed infection due to lineage 3, 4, or lineage 2, 3, and 4. Northeastern region reports more of BJ. So infections due to lineage 4 were reported more during and after the COVID-19 pandemic. Now, what I was alluding to earlier, lineage 4 has not been reported from our country in a big manner. It is primarily a lineage of the Western world. Some are reported from Russia, but primarily the whole of Europe, North America, South America reports lineage 4. And what we found distinctly that lineage 4 was responsible for polyclonal infections in sensitive TB, while it was responsible for MDR TB clonal infections in significant number of cases. Now, this is just to bring in a recent article by Freshy et al., which reports that lineage 4 has shortest median terminal branch length as compared to the other, global lineages 1, 2, and 3. Hence, it has higher transmissibility. They've also shown internode to tip distance is another phylogenetic correlate of transmissibility. The distribution of this measure across the 4 lineages also reveals a similar hierarchy. So the purple one being lineage 4. So to conclude, the COVID-19 pandemic actually appears to be responsible for an epidemiological change in the prevalent genotypes of tuberculosis in India. Increasing number of polyclonal infections are likely to be associated with difficult to treat TB as well as relapses. Highly transmissible lineage 4, which contributed to a small proportion of TB cases in India, appears to be spreading as well as causing polyclonal infections. So there is more work going on. This was using the SNP genotyping method. We are now doing whole genome sequencing to see if our results from the SNP typing method are replicated in the whole genome sequencing studies. And this is thank you from South Africa, obrigada from Brazil. I don't know how to pronounce that, from Russia, and dhanyavad from India. And thank you all for the patient listening. I can take questions. If there are any questions, Dr. Bimal can also take the questions. I think we were too explicit.
So I think I'll thank both Dr. Bimal and Dr. Urishi at the last minute. They were able to at least honor our commitment for the CGR. So I'm really thankful to them. Any questions, then we can ask. Otherwise, thank you. They are there. You can catch them anytime. Very interesting. It's very nice. Very good present. Yeah. Thank you.